My name is Tititip Tipiyamontri. I'm from University of Sherbrooke, Quebec, Canada. I'm a postdoc student, and I'm here in the radiation research meeting, and I would like to interview Dr. Sijumon Kunjachan. Hi, could you please tell us a little bit about your research?
Sure. So my name is Sijumon Kunjachan, and I work at Brigham and Women's Hospital at Harvard Medical School. I work on nanoparticles for radiation therapy. My main project is focusing on understanding the potential of nanoparticles to induce tumor vascular disruption followed by radiation therapy. And this is a very new area of research, and it's completely different from the conventional way of treating with nanoparticles, where nanoparticles are used to target the tumor cells first and irradiation is applied onto them. There are several barriers which stop nanoparticles from reaching the tumor cells in certain cancers. For example, in pancreatic adenocarcinoma, we have a dense stromal matrix which restricts the flow of nanoparticles from blood vasculature to the tumor cells. So in those cases, it's very difficult to target the tumor cells. Therefore, we adopt a new technique where we can use nanoparticles to localize them on tumor vascular walls and then use radiation to damage the tumor vessels. That's the main focus of my current research.
Perfect. And can you tell a little bit about challenging of doing this research? I know it's very challenging to work with nanoparticles and other stuff.
Right. So nanoparticles per se have several disadvantages when compared to other chemical molecules. Primarily, nanoparticles are taken up by macrophages soon after injecting into the mice. This is partly because of the opsonization process that happens. To prevent this, we have to make sure that the nanoparticles that we synthesize have hydrophilic coating on them, which can avoid the nanoparticles being recognized by the macrophages, and it can go to the tumor in large quantities. And localizing them at tumor endothelial surface is a bit more difficult because we have to have a peptide on the surface of the nanoparticles, and we should make sure that they are stable in the bloodstream so that we can find out a point where maximum number of nanoparticles accumulate in the blood vessel, and we can induce radiation to cause damage to the vasculature.
And what about the size of nanoparticles that you use?
The size is very critical, primarily because, like I previously said, bigger size nanoparticles are heavily taken up by macrophages. A size dimensions of less than 5 nanometer. In my case, I'm using 1 to 2 nanometers.
Very small.
These are very good in the sense that these are less recognized by the circulating macrophages, and they're likely to end up in the tumor well. And secondly, size also decides where and how much it can diffuse through smaller blood vessels, like microvasculature, which are critical parameters in radiation, because radiation has a major impact on tumor microvasculature.
Very nice. And once you observe your results, it's very interesting results, I have seen that. How you translate your results into other fields, such as in radiation research, in clinical radiotherapy? Can you please tell us about that?
Right. So in the clinics, we usually use chemical vascular disrupting agents. One of the main disadvantages of clinical vascular disrupting agents is that it distributes everywhere in the body. It can affect all visceral organs, starting from liver, spleen, kidney, and so on. There's no control over where it goes. Wherever blood flows, these chemical vascular disrupting agents goes. The advantage with nanoparticles is that we can regulate and modulate its accumulation specifically in the tumor.
Targeting only in the tumor.
Only in the tumor. And let's say some amount of it goes to the liver, but since the radiation is target specific, it doesn't activate the nanoparticles which are in the liver, which means this is kind of a dual targeting system where nanoparticles are specifically activated on the tumor, and it can induce a therapeutic efficacy. In translational, when we think about the translational part of it, biocompatibility is a main issue. We have to make sure that these nanoparticles are not toxic per se. And previous research have already shown that nanoparticles, particularly gold, doesn't have a biological activity. It's more or less inert. And in the moderate quantities that we use, it's fairly good in terms of its toxicity. And it doesn't cause any adverse reactions as such. And recent studies also show that gold nanoparticles eventually gets cleared out of the body, and there's much less accumulating in other organs, but this happens over time. So from a clinical perspective, this is actually very good. If we can have a nanoparticle in the tumor, induce a damage, making sure that rest of the organs are protected, and the contrast agent, which is the nanoparticles here, if they are cleared out of the body per se, these are very good because it is less toxic and it is high therapeutic efficacy. So that's the main idea behind it. So my work focused on using a small animal radiation platform, using a low energy beam, and try to look into the concept of vascular disruption. And we actually did that and showed that. So one of the posters which I presented for this conference is about tumor vascular disruption. We clearly showed for the first time that tumor vascular disruption can be induced if we use nanoparticles in optimum amounts and localize them to the tumor endothelial walls and use radiation. From a clinical perspective, we might have to go for higher radiation strength, which means we'll have to test them with a 6 MV, which is something what we are planning to look forward to. And we'll be testing them in higher radiation doses.
And you can say your gold nanoparticles can behave like a radiosynthesizer.
Yes, so gold itself is a high molecular weight metal, which can improve the radiation, and it can bring forth radiation enhancement.
Yes.
And therefore, it's much preferred over other metals.
Okay. I was wondering and curious to know what's the concentration of the gold nanoparticles that have to be in the tumor and behave like a radiosynthesizing?
Okay, that's a very good question. So previously, people used gold nanoparticles, and the mechanism was passive tumor targeting, which means gold itself pass through the tumor blood vessels, leak through the leaky vasculature, reach the tumor cells, and then induce a radiation effect. So in that concept, it's important that we have a heavy payload of tumor cells being delivered with gold nanoparticles. So the percentage injected dose or the fraction of gold nanoparticles that reach the tumor is very important. But in our approach, we majorly focus on vasculature, primarily because if you can shut down the vascular supply, you can induce damage to a multitude of cells which is being supplied by this blood vessel. So in this case, we probably might not need so high concentration as much as we need for doing tumor cellular targeting, but we definitely need optimum amounts across the endothelial walls. So the dose that we use is way lesser compared to the previous reported dose on tumor cell targeting, which is like 1.2 milligram per gram. And therefore, we avoid high toxicities.
Okay. And when you damage the vascularization, you reduce the oxygen's flow.
Right.
You might create hypoxic area in your tumor.
Right.
How can you explain this?
That's a very good question. That's a major challenge in vascular disruption. It might cause hypoxia, which is anyways innate. But how much this hypoxia will affect other therapies, that's the main thing, and how much it will affect chemotherapies, which will be administered along with this. That's a major question, and one of the projects that we are working on currently is to understand how much is the hypoxic change pre and post vascular disruption. But certainly, that's the main downside. If it cannot be solved, if it cannot be combated with our methods.
Yes.
Right.
So this is going to be your developed future.
Exactly.
Research.
That's a parallel project that we are starting to work on, to understand how much it contributes to increasing hypoxia, or in a way it might be also helpful in the sense that these hypoxic cells might turn completely necrotic, and eventually, but it depends on the heterogeneity of the tumor, and it's highly dependent on the tumor model that we look for.
So what type of tumor that you are working on now?
Right.
So this is the question. Yes.
Very good question. So I work on pancreatic tumor model. Why I chose this model is simply because, unlike other tumor models, if we inject gold nanoparticles, which usually leaks through the blood vessels and go to the tumor cells, in pancreatic tumor model, they have very dense extracellular matrix, which means these particles, even if they diffuse, they are not farther beyond the tumor blood vessels, but they are more or less concentrated on the endothelial walls, which is actually a boon for our therapy, because in that case, we would anyways irradiate them and damage the vasculature. So pancreatic tumor model is an otherwise notorious model where this therapy could be a very good advantage.
And you think this can be like future close to the clinical trials?
Oh yes, we are primarily testing on a 6 MV beam, and if that shows very promising results, we'll certainly work towards its clinical translation possibilities.
So very good. So I have a little bit concern about imaging again. So you did a lot of work, very great work in imaging using nanotechnology.
Right.
Can you comment and give us some, like information about using nanotechnology in imaging world?
Please? Right. So nanotechnology is a very good platform to have imaging and radiation to be performed at the same time. Unlike injecting an imaging agent and doing imaging separately, we could use a single agent and we can induce a radiation effect and at the same time visualize them. So one of the projects that's going on in our lab is mainly focusing on how we can use these kind of dual systems which can induce radiation and which can be used for imaging, and how we can look into them. So these are the novel theranostic models that we are trying to look into. So the agents, if what we inject, if they can give a contrast in an imaging modality, then we can really see them as we induce the radiation. So we are looking into several gadolinium-based nanoparticles which can bring radiation and which can also be which can be used to perform imaging at the same time, so that it can be like a theranostic model which can be used for theranostic applications.
Very interesting. And if you can say us a little bit again about the application of your research in other field, like I will repeat this question because in this meeting we have many people from different fields, such as from immunotherapy, from people from the space radiation. Can nanotechnology develop or apply to the other field?
Right. It has a great amount of influence on other fields. Primarily, immunology is a developing field with respect to this aspect. So if we look into what happens after tumor vascular disruption, that like with respect to what will happen with respect to the immune system, how will the immune system react with respect to tumor vascular disruption, that will give us more information how we can use immune system to act against the tumor using tumor vascular disruption. So that's certainly a very interesting field. And one of the other things which we might be interested in is like to understand how much we can use this strategy to overcome multi-drug resistance in tumors. So multi-drug resistance is predominantly a surface characteristic of the tumor cell, whereas this is a mechanical application of radiation. So there might be ways by which we can overcome multi-drug resistance using gold nanoparticles in radiation therapy. That's something we are looking into.
Okay. You work now mostly in preclinical? Or
right.
You use the mouse model?
Yes. We test them in different pancreatic models.
Yes.
What would be further?
Autotopic implantation of the tumor cell into the pancreas?
To start with, we actually look into subcutaneous models. Simply because we wanted to prove the concept that we can induce tumor vascular disruption upon irradiation using gold nanoparticles. And now that we have seen it doing the disruption, we plan to look into other tumor models, autotopic and several other clinically relevant tumor models.
Very nice. Yes. If you want to challenge yourself in the future, not your planned future developing research, but if you want to challenge yourself in research, radiation research field, what the subject that you want to challenge? What you want to, progress or do some difference or try to find a solution? Right.
I'm very much interested in knowing how radiation by itself can be a monotherapy, and how it can also trigger other things like, for example, using a nanoparticle platform to do radiation at the same time to do chemotherapy by delivering drugs on the same platform, which means we can induce a damage we can also facilitate these endothelial damage to bring in more drugs to the tumor. So this is kind of a synergistic approach, and that's what I like to see more in the coming days. Expanding on it, like we have more drug delivery plus radiation happening with this nanotechnology platform.
So could you please tell us how you take the advantage of or benefits of attending this radiation research meeting?
Okay, this meeting was a great platform in the sense that I was able to meet people from different diverse backgrounds who are still working with radiation, but they also have something else on their side, like an imaging or some kind of immunotherapy. So this was great in the sense that when we discussed with them, it just gave a lot of ideas as to how to do particular thing. I've had a chance to meet several people during my poster session and discuss with them several possibilities of tracing the damage, the vascular damage that we induce. Many of them gave great ideas, and that's particularly very interesting in this research conference.
Yes. So I would like to thank Dr. Sijumon Kunjachan very much for your time and very great knowledge in the field that you have been working on, and very interesting clinical study that can translate from preclinicals into the clinical world, and is very important for us. Thank you very much.
Thank you.
Thank you.